this presentation, I will discuss enzyme-drug interactions and the molecular mechanism of how these drugs target their cognate enzymes. Specifically, I will describe mechanism-based inhibition of HIV protease, aspirin, acetylcholinesterase inhibition, and penicillin. These are all enzymes that use covalent catalysis in their catalytic mechanism, similar to serine proteases like chymotrypsin. The first drug I will describe is aspirin and how it inactivates the COX-1 and COX-2 enzymes. COX-1 and COX-2 are difunctional enzymes, meaning that they have two catalytic activities. They're both cyclooxygenases and peroxidases. COX-1 and COX-2 are involved in the synthesis of prostaglandins from phospholipid precursors. Prostaglandins regulate many physiological processes, including platelet aggregation, uterine contraction, pain, inflammation, and the secretion of gastric mucosa that protects the stomach from gastric acids and proteases. Aspirin and ibuprofen are inflammatory drugs that prevent prostaglandin production by inhibition of the COX enzymes. In its mechanism of action, aspirin irreversibly inactivates the cyclooxygenase activity of COX-1 and 2 by acetylating a serine residue. This blocks the enzyme's active site and inhibits the production of prostaglandins. Ibuprofen is another anti-inflammatory drug, but differs from aspirin by its mechanism of action. Ibuprofen inhibits COX enzymes by binding and mimicking an intermediate in the reaction, but does not chemically modify COX as does aspirin. So ibuprofen is actually a competitive inhibitor of the COX enzymes. Every year, thousands of tons of aspirin are consumed for the relief of headaches, sore muscles, inflamed joints, and fever. Unfortunately, aspirin can have serious side effects including stomach bleeding, kidney failure, and even death. In turn, drug companies have attempted to develop a better aspirin such as ibuprofen, acetaminophen, and naproxen, with the goal of reducing these side effects from COX-1. In mammals, there are two COX enzymes, COX-1 and COX-2. COX-1 is responsible for the synthesis of prostaglandins that regulate the secretion of gastric mucosa. COX-2 is responsible for the synthesis of prostaglandins that mediate inflammation, pain, and fever. While these enzymes have different functions, they have 60% sequence identity and have similar reaction mechanisms, making it very difficult to specifically target just COX-2 without the side effects of targeting COX-1. Vioxx is a COX-2 inhibitor and is a story of a good drug gone bad. The active ingredient of Vioxx, rofecoxib, is a selective COX-2 inhibitor and was approved as safe and effective by the Food and Drug Administration. It was used to treat acute pain and arthritis and actually worked very well for its prescribed treatment. However, in September 30, 2004, Merck withdrew Vioxx from the market because of safety concerns about risk of heart attack and stroke and death associated with long-term high-dosage use of Vioxx. There are claims that Merck knew of these safety concerns and concealed this information, which is not surprising given that in 2003, the year before the withdrawal, Merck had a sales revenue of $2.5 billion from Vioxx. It remains unclear why Vioxx caused cardiovascular malfunction, but since then, the development of COX-2-only inhibitors is a subject of controversy. Dispropylphosphofluoridate, also called DIPF, is a neurotoxin that irreversibly inactivates acetylcholinesterase. Acetylcholinesterase is an important enzyme involved in terminating and resetting nerve impulses. It hydrolyzes the neurotransmitter acetylcholine, utilizing a catalytic mechanism that is very similar to the serine proteases. DIPF, which is not a drug, and similar compounds like sarin, are the active ingredient in military nerve gases. The mechanism of DIP inactivation of acetylcholinesterase involves the formation of a drug-enzyme covalent intermediate via an active site serine. This covalent intermediate inactivates the enzyme, which in turn causes acetylcholine to accumulate, and nerve impulses cannot be stopped, causing prolonged muscle contraction leading to paralysis and even death. Penicillin and its derivatives are a group of potent antibiotics used to treat antibacterial infections. Penicillins have a thiazolidine ring, a variable R group, and a reactive peptide bond in a beta-lactam ring. Penicillin kills bacteria by inhibiting the bacterial enzyme glycopeptide transpeptidase, which is involved in cell wall biogenesis. Cell wall biosynthesis requires the cross-linking of a terminal glycine-pentaglycine bridge with a terminal D-allo-D allo unit, which is catalyzed by glycopeptide transpeptidase to form the Gly-D-allo crosslink. The first step of this reaction involves the initial formation of a D-allo-acyl enzyme intermediate. In the second step, the acyl intermediate reacts with the Gly-containing pentaglycine bridge to form the final crosslink product. The reactive bond of penicillin resembles the bond formed in the transition state of the initial D-allo-D-allo substrate reaction. In the mechanism of action, penicillin forms a stable covalent adduct with the transpeptidase and inactivates it. Unfortunately, a major problem in antibiotic resistance is due to the fact that bacteria produce beta-lactamases that destroy the penicillin ring and counteract the transpeptidase inactivation. HIV contains an essential aspartyl protease that is involved in cleaving multi-domain viral proteins into their active forms, which is necessary for virus reproduction. This enzyme is called HIV protease. Aspartyl proteases are proteases that use an active site aspartic acid residue to abstract a proton from water so that it can better nucleophilically attack the carbonyl carbon in a peptide bond. The crystal structure of HIV protease revealed that HIV protease is a homodimer in which two monomers form the active site. Each monomer contributes an active site aspartate, and the active site is a narrow tunnel covered by flexible flops that open and close to allow for binding and release of products and substrates. Knowledge of the mechanism and structure of HIV protease has led to the rational design of HIV protease inhibitors used in the treatment of AIDS. One of these drugs is called Crixivan, and it contains phenyl and other bulky groups that bind to HIV protease in the active site tunnel. Of greater importance, Crixivan has a correct geometry to mimic the tetrahedral intermediate of the substrate. In the active site tunnel, Crixivan interacts strongly with the aspartic acid residues. Crixivan also causes the flaps of the HIV protease to lock into place. Together, these properties make Crixivan a high-affinity transition state analog that locks up HIV protease and prevents it from binding and cleaving proteins required for viral particle production.